Hi everybody. Welcome back to my series of lectures on protein sequence databases. Today I want to expand a little bit more on UniProt, which is the main protein sequence database, as you might recall from Tuesday's lecture. And I also want to look a little bit more in depth at some of the information inside UniProt KB. So if you remember, UniProt KB was the main repository, but UniProt had some other databases as well. But we're going to focus today more on the UniProt KB side and delve a little bit more into some of the philosophy of why the database is the way it is. We're also going to spend very few minutes talking about sequence motifs, just because I think it's an interesting aspect of protein sequence databases in that sequence codes for sequence motifs. And sequence motifs are extremely important in bioinformatics. So I've prepared a slide on that. We're also going to slowly get into the mindset of thinking about protein structure. So as a bioinformatician, I'm obviously very interested in protein sequence. But protein sequence is one important cornerstone of protein structure studies. And we're going to be discussing protein structure in great detail next time I speak to you, and we have 2 lectures scheduled for that, but I did want to get us a little bit into the mindset of thinking about protein structure. Okay, let's get started. So this is our schedule of events for the day. I want to do a bit of recap on UniProt from last lecture on Tuesday, because I think it's important for us to spend a bit of time revisiting that issue. Now, I'm not sure if you had any questions for me about last week's lecture, but I still thought it was better to err on the side of caution and spend a bit of time looking at a couple of slides from last week. Next, we're going to look at the constituent databases of UniProt, UniProt KB in particular. Now, just a note, if I interchange UniProt and UniProt KB, it's because generally people who talk about UniProt are referring, in fact, to UniProt KB. So don't be confused if I switch back and forth. It is a bioinformatics term, UniProt, and UniProt KB is the main database that gives us the term UniProt. So I'm using both terms equivalently, unless I specifically mention one of the other databases inside the UniProt family. But let's spend a bit of time dividing and conquering UniProt KB today. Then I want to spend a few minutes talking to you about what it means to manually curate a database and some of the metadata that's available to us inside UniProt. And fourth, as I mentioned, I want to get us thinking, as you go home this weekend, I want you to start thinking about protein sequence databases in the context of protein structure. We're not going to get into any of the biology or the bioinformatics of that today, because we have 2 lectures devoted to that, and I want to really spend some time hammering home some points about the basic biology, because you have to understand the biology in order to be an effective protein bioinformatician. Finally, before we end off, I'm going to very briefly spend a few minutes discussing sequence motifs with you, because they're very interesting to bioinformaticians, and there are a lot of studies in place to understand sequence motifs, so I thought I should give you a brief overview on that. And then we end for the day, and in fact for the week. Okay, so the next 2 slides are taken from last week's lecture, but I wanted to structure my talk around them because they are still very critical to this lecture and for any other work you will be doing with proteins, both in this course and in your potential future career as bioinformaticians. So, UniProt is composed of this big database called UniProt KB, which contains all the protein sequences and some other databases. Now, I showed you a screen capture last week of the UniProt homepage, which had in it the UniProt database, UniPark, UniRef, and all the other resources available to you when you're accessing the UniProt repository. However, UniProt KB is the one that everyone is generally interested in when we say UniProt. UniProt is a collaboration between 3 projects. As I said, one of them is next door here in Cambridge, the EBI. One is the Swiss Institute for Bioinformatics, and the other one is the Protein Information Resource, which is indeed an American collaboration. I did make sure I looked it up afterwards so I could present that information to you. And just as we discussed in our networking lecture, all these databases that are principal repositories for their particular domains, so UniProt is for protein sequence, GenBank is for DNA RNA sequences, PDB is for protein structure, and PubMed is for publications. These big databases really like to talk to each other, so there's always cross-referencing going on. If there's a publication about some protein sequence that's been discovered, there will always be a link to the UniProt accession number or the accession ID or the retrieval ID or the unique identifier for that sequence inside UniProt. And likewise, GenBank will reference PDB and PubMed. PubMed will reference everything. PDB will have links to the source UniProt sequence. That is actually very important, and we will see why that is when we start looking at protein structure. And once again, 95% of the data inside UniProt KB comes from the translation of DNA sequences that are submitted to GenBank. So GenBank gets a DNA sequence which has an ID, and then that sequence is retrieved by the automated bioinformatics scripts inside UniProt, and the manual curators who work tirelessly to ensure quality, and they are converted to amino acid sequences and deposited into UniProt KB. Now, if you remember, we ran a small Python script last lecture, and you got to see a little bit about how this kind of translation can be done very quickly in a few lines of code. And again, here's the About page, which I again highly recommend that you go and look at the About pages of all of these databases because it tells you some very interesting history. UniProt, each sequence inside UniProt has a certain category. It's called a protein existence score, where PE1 is definitely we have experimental evidence to verify this protein, and PE5 is usually some sort of hypothetical protein or a translated sequence from some very low quality DNA. This happens a lot with fossils, for instance. If we're looking at the fossil record and recent fossils will still have traces of DNA, and it could be very badly damaged, but we could still possibly get a putative amino acid sequence. Those are generally given scores of PE4 or 5, depending on how badly damaged the DNA was initially. And here's the link to go read about it. Again, remember that you can click on these links to take you directly, and it should open up your web browser. Okay, now UniProt KB itself is composed of 2 constituent databases. Now I'm going to pull up a web browser because I want to show this to you. So if I go to my Firefox and I go to uniprot.org So this is the homepage for UniProt, and as you can see, UniProt KB is composed of this one box called SwissProt, and the other one is called Tremble. Okay, so those are the two constituent projects inside UniProt. SwissProt was developed in 1986. That was the first project. Before this umbrella UniProt existed, it was its own project, and as the name may indicate, it was based in Switzerland. And it is very high quality, it's manually curated, and the sequences are non-redundant, which means that identical sequences will not occur in SwissProt. And the redundancy is removed through manual curation, which means human beings actually sit down, look at the sequences, and do a bit of work to determine the quality and which sequences stay in the database and which sequences get kicked out. And it's composed of experimental results, so sequences that have been determined experimentally through mass spec, sequencing, and the in silico translation. So the DNA that gets sequenced through a sequencing facility can then be translated. In silico just means inside a computer. In silico is Latin for inside silicon, which is what is inside your computer chip. So in silico translation of the DNA to produce the amino acid sequence, combined with experimental results to verify these proteins, is what SwissProt is composed of. A much newer project started in 1996, 10 years after SwissProt, and that was called Tremble. The EMBL in Tremble comes from the European Molecular Biology Lab, okay? It's one of the largest bioinformatics and biotech and biology institutes in the world. It's based in Europe. There are many different sites, one of which is the EBI, the European Bioinformatics Institute, which is based in Cambridge. And Tremble is automatic, so it is composed of a whole bunch of Python scripts and Perl scripts and C++ code that sit there waiting for new DNA and RNA sequences to be entered into GenBank. And as soon as GenBank gets a new release, Tremble will pull out all the new sequences and automatically annotate as many of those sequences as it can. And Tremble was initially designed because SwissProt was just a bit slow and it couldn't keep up with the amount of sequencing data that we were producing. Now, does this mean it's a bad thing? Well, I'll leave you to think about that, but basically what you want to think about with SwissProt and Tremble is that they are kept separate, so SwissProt sequences will never mix with Tremble and vice versa. And so if you are designing a bioinformatics experiment to study protein sequence, which one of these would you use? And which is of higher quality? And does using one mean that the other one is useless? Or does it mean that they both have different uses? So think about that. It's a philosophical question. It's quite open-ended, actually. The answer may not be as clear as you think it is. And indeed, bioinformaticians will decide which one of these repositories to use depending on what they're actually trying to achieve. So let's take a look a bit at manual curation or manual annotation. So inside SwissProt, the first step is that the sequences get curated. So the new DNA RNA sequence that the scientists have decided to study for incorporation into UniProt KB is chosen, and it is blasted. So the blast algorithm is run on this sequence to find homologous sequences. So again, homologs by definition are sequences which have some evolutionary relationship with each other. So they're similar. The second step is that these sequences, or this chosen sequence, is then put through a rigorous analysis. So there are different algorithms that look at post-translational modifications that could happen to this amino acid sequence, where in the cell this protein is likely to occur, what kind of sequence domains it has, what kind of motifs it has, which we will come to later, etc. Now, before I go to number 3, actually, I just want to point out that a lot of these annotations and curation steps do make use of software. The point is that the manual curation happens because scientists actually are very closely tied to the software. They're constantly analyzing the output, and they are deciding on whether a certain annotation should be given to a certain protein sequence based on what the software says. So just because it's manual doesn't mean that there's no software that's used. I just mean SwissProt is manual when compared to Tremble, which is entirely automatic and has no human intervention. All right, number 3. Thirdly, a literature search is done. So relevant journal articles to this sequence are located in PubMed, and these are then pulled out to add to this metadata that we already have from steps 1 and 2. So remember that all these bits constitute the metadata. The actual data is just the sequence. Then fourthly, there's some more study on homologous sequences, and they look and see which potential protein family this sequence could go into just based on sequence similarity. So if we know that there's a family of proteins that is responsible for unwinding DNA during DNA replication, for instance, bonus points if you can tell me what this family of enzymes is called. But that could be a certain family of proteins. So if you've got a novel sequence that comes in, perhaps there is a hit to this family of proteins, and then they can say, well, okay, as part of the metadata, we're going to put down that it looks like this unknown sequence could be a protein of this family. By the way, that family of proteins is called helicases. So because DNA is a helix, the enzyme that unwraps the helix is called a helicase. Then there's a quality assurance step. The curation is then thoroughly checked again by different scientists at different levels. There's a lot of paperwork that goes on to make sure that these sequences can then get incorporated. It's signed off by some of the principal scientists at these facilities. And then finally it's available in the UniProt KB SwissProt section. So this is sort of the step that each protein sequence that's novel gets to go through in order to be evaluated for entry into SwissProt. So it's a very rigorous process. Now, I hope you understood and took home the message from this slide that metadata is extremely important. In all of these steps, almost everything that we're doing is just adding to the metadata, to the knowledge that we have about this unknown sequence. So my question to you is, why are bioinformaticians obsessed with high-quality metadata? It really does become an obsession sometimes. So think about why that may be. It's not just about, well, we have some data and we know which species it comes from. Why are we so interested in looking at all this different kind of information that we can attach to this sequence? It's just something for you to think about. So right now we're going to do a little bit of a recap and think back to our lecture on networks and databases. And we're going to just do a quick study here, okay? So the entire UniProt database from the home page, you can download it, okay? And it's updated every 4 weeks. So again, in a bioinformatics facility, normally there's a script that checks every 4 weeks and downloads the new UniProt database, okay? Very simple. So here's some information for you. The SwissProt section of UniProt KB, which is available as its own archive, is about 480 megabytes. When I uncompress it, it is about 2.8 gigabytes, and this is the plain text format, so there's not even any FASTA headers or any of that. It literally is just a list of sequences and all their metadata or annotations. How much smaller is the archive? This should be pretty easy for you to do. It's 2.8 gigabytes divided by 480 megabytes, and the number you get will tell you the difference between the archive and the uncompressed size. Okay? Oh, make sure you keep your units the same. So 2.8 gigabytes is 2,800 megabytes. Now this question is a little bit trickier, but I put it on here because I think it's a useful exercise. If the average protein or amino acid sequence contains 250 amino acids, and the average annotation for a protein, so all the metadata that you have for a protein is approximately 4,800 characters long, approximately how many annotated entries do we have in SwissProt? Now, you can go to uniprot.org, and on the homepage, you might have even seen it when I pulled up the browser, that it told you what the answer was right there. But this is a case where if we know that the average protein is approximately so long, and we know that the approximate annotation is that big, we can make a good guess for how many annotated entries we have in SwissProt. And just as a little hint, you're going to need this number here, this 2.8 gigabytes. Okay? And just to compare, this number should be a lot smaller, and I will show you what this number is in a few minutes. But just to give you an idea, the UniProt KB Tremble side of things has over 82 million sequences. All right. Now I want to switch focus and discuss how UniProt is actually the gateway to structure. Okay, so remember, UniProt is a sequence database. So why am I talking about protein structure? So as you've seen, UniProt is very useful for basic scientific understanding. Anytime there's a new DNA or RNA sequence that codes for a protein, you're going to find that protein sequence somewhere in UniProt. It's either going to be in Tremble or it's going to get curated and go into SwissProt. Okay? So it increases our understanding of basic protein sequences. That's great. That's very important. What else can we do with it? So it is the de facto repository. So it is absolutely the default repository that everyone's going to go to when they're looking for reference sequences, when they're determining structure. So the determination of protein structure is not something I want to get into in great detail today, but I will certainly give you more details about this when we talk about protein structure. But remember that protein sequence, to some extent, influences the protein structure. And why is that? Well, think back to your biology. What are proteins composed of in their sequence? Amino acids. Think about the chemical properties of amino acids. Think of what kind of charges each of these amino acids has. Okay? And so when differently charged amino acids come together in a liquid environment, they're going to fold, and they're going to induce protein folding. And it is the folding of the protein that is what we refer to as the structure of the protein. And we're going to get into much more detail about this later. But the sequence does in many ways affect the structure of the protein. So when you have a de facto repository for protein sequences, you can certainly use them and use bioinformatics algorithms to determine something about the protein structure. And we'll come to this later. So the reference sequences that you can get out of UniProt can be used by structure building algorithms to determine the 3D shape of a protein. Now this is a very simplistic statement. It's actually not nearly as easy as I've just described it. But we're going to look and see what tools we have available to bridge the gap between sequence and structure. And just when you look at these slides later on, if you're wondering where all the information on protein structure is, I've just put that there so you know that it's coming up later. Okay, very quickly before we end off today, I want to discuss sequence motifs. So what is a sequence motif? Well, what is a motif in general? It's a short, recognizable stretch of sequence that occurs in different proteins that occur in different species, and motifs are recognizable across different organisms within a species. So different portions of proteins and protein sequences are conserved within different species. So you can have a short stretch of protein sequence that's very common between two related species. Conversely, if you have two species and you don't know if they're related, chances are they're going to have a lot of sequence motifs in common if they are related. Very quickly, think back to your bioinformatics. How do we determine commonality between two sequences? We conduct sequence alignment. And how do you compare one sequence against a large database? Well, sequence alignment, but more specifically, we like to use the BLAST algorithm. Now, the key is, why would biology... Now, this is something you want to keep in mind whenever we talk about conservation of sequences or structure. It's not just by random chance that this happens. I mean, yes, evolution and random mutations do end up leading to unique sequences and structures. But evolution is very good at removing sequences and structures that do not help the species survive. So chances are, if you have sequence motifs that are common today, that are evolutionarily conserved, they must have a biological significance. Something about that sequence is keeping that animal alive. Okay? That's the bottom line. That something about that sequence is keeping that plant going. Something about the sequence motifs in the proteins of alligators made them such a successful species that they survived the dinosaur extinction. So whenever we're talking about sequence motifs and all these bioinformatics-y kind of computational words, keep in mind that there's always a greater biological significance to these things. Keep the biology in mind. And indeed, they do have certain functions. So short, recognizable stretches of sequences are not just recognizable to you and me, but they're very recognizable to other proteins that perhaps participate in DNA binding. Perhaps there are sites for glycosylation. Maybe there are sites for methylation, different modifications that the protein goes through once it has been translated in order to activate it or in order to suppress it. In other words, regulate the protein. So different proteins will come together and find and recognize these short stretches of sequence in the target protein and then do something to it. These are only 3 functions. Sequence motifs have many, many more functions in biology, but I wanted to give you 3 important and very simple examples of what some of these functions could be for motifs. And how do we computationally discover them? Multiple sequence alignment, clustering, BLAST, local sequence alignment. However, when you have an unknown sequence and you're trying to compare it against a group of sequences, you're not just going to run a simple sequence alignment. You're going to be running multiple sequence alignment, BLAST. You're going to cluster these things together. Now, just a point here, multiple sequence alignment is very good for determining conserved sequence motifs across multiple sequences. So, in general, you will tend to use a multiple sequence alignment approach if you're looking for sequence motifs. Okay? And so, where do we get our information about sequence motifs? Well, UniProt KB is the biggest resource for protein sequences, so quite naturally it's going to be the biggest and the best curated resource for sequence motifs themselves. So think of sequence motifs and the study of sequence motifs as one of the most direct applications of using UniProt KB. And indeed, people are obsessed with sequence motifs. And they're very, very strongly implicated in a lot of diseases. And as we understand more and more about them, we can use this to create drugs against these sequence motifs. We can use it to regulate these proteins. I mean, if some of these motifs are regulatory by nature, can we leverage the machinery of the cell itself to regulate some of these proteins, perhaps prevent some cancers, perhaps even work as antivirals? So, sequence motifs are a very direct application of UniProt KB at the sequence level without even getting into the structure. Okay, so I thought this would be a good example for you to understand a real-world bioinformatics application for UniProt KB. And continuing that theme, what kind of metadata does SwissProt have that would help me identify whether two unknown sequences are related by common motifs? Now, I'm deliberately keeping this question vague, and I am deliberately not asking you to answer this question now, apart from the fact that we're on YouTube. What I want you to think about here is spend some time on the UniProt website and look and see what kind of metadata we have that could maybe tell us something about the sequence motifs that are present in a particular sequence or in a set of two sequences. But think of what other metadata we could look at besides the sequence motifs themselves, okay? So remember I said that two sequences that come from related species will often have common sequence motifs. So think about working top-down. So there are two approaches here. The bottom-up approach is that you have the two sequence motifs and you're trying to discover what organism it is. The top-down approach is what I'm asking here, which is you have two organisms, for instance, and you have two unknown sequences, and you're trying to determine what metadata you could use inside SwissProt to determine whether these sequences are related by sequence motifs, and if so, what those organisms could be. Again, this is not an exercise in programming or any of these things. It's a question for you to answer just by looking at the SwissProt website and navigating through and looking and seeing what kind of data you get. Now, again, I do believe that you'll be spending a lot of time with UniProt in your labs, and I think this might be a good question for you to think about answering. Just an exercise. So before we end today, as usual, I like to go through a little summary and talk about what we have just spoken about. Now, this lecture has been a little bit shorter. Pardon me, I'm just going to take a sip of water. This lecture has been a little bit shorter because I spent a lot of time last week looking at the demonstrations for Python and looking at the UniProt website, and I wanted to give you some more theory today. So for people who don't like theory, I do apologize. I was never a big fan of theory back in the day, but as I got an appreciation for some of these resources we have available, I became very interested in understanding how these databases were built. So that's what we've been trying to do today. So remember that UniProt KB is composed of two big databases that do not talk to each other, SwissProt and Tremble. I'm going to ask some questions as we go through these points as well, just because I think they're good review material for you. So my first question is, which database is bigger? Well, Tremble. Tremble has about 82 million sequences. SwissProt has 500,000. Now, does that necessarily mean bigger is better? So why was Tremble built? Tremble was built to address the need where there were many, many more DNA sequences being produced and discovered, and so SwissProt and the scientists couldn't keep up with annotating these sequences. So Tremble was developed as a way, almost an intermediary, where some of these sequences could sit until the SwissProt scientists got around to them, or better annotation tools became available. But is bigger better in this case? Is it just good that we have a huge database of proteins? Tremble isn't bad. It does go through a lot of automatic annotations. But I will pretty much say that SwissProt is the database you want to be using. Just because we know that the quality is going to be very, very good. And so you're less likely to make mistakes if you use data from SwissProt than from Tremble. So, following on from that, manual curation makes SwissProt the better resource to use in almost any application. So if you are writing bioinformatics algorithms or scripts, that need to work on UniProt KB sequences, maybe you're doing a survey of the proteins in UniProt, I would advise restricting yourself to SwissProt. But on the other hand, when might Tremble be useful? So what's the point if you have 82 million sequences? It's great that we have all these sequences, but there's no point if nobody uses them. Well, I will tell you right now that many people use Tremble. And so when might Tremble be useful? So think about that. Also today, we gained an appreciation for the size of SwissProt. So again, to recap, it was around 560,000 odd sequences. Now, here's a quick reflection from an earlier slide. How often does a new release come out? The answer is about 4 weeks. Okay, so SwissProt is released every 4 weeks. And again, all this information is available on UniProt's website, and I probably alluded to it earlier. And if I didn't, well, I'm going to say it again. It's about 4 weeks that you get a new SwissProt release. Metadata. We've spent some time looking at some of the metadata today in SwissProt. It is more important than data in many respects. I strongly believe that it is more important than data. Because if you have a sequence, it's great you have a sequence. If you don't know anything about it, well, it's just a string of letters. So, from the last lecture, can you name 2 kinds of metadata that are stored in UniProt KB? So if you remember, we looked at the UniProt homepage, and I pointed out distinctly some different kinds of metadata that we had available to us that we could search on, that we could classify, that we could cluster. So can you name 2 kinds of metadata? It's on the homepage. Then we got into the mindset of transitioning towards protein structure. And quick recap, what is the de facto repository for protein structures? So UniProt is the repository for protein sequences, and the repository for structures is known as the Protein Data Bank, or the PDB. We're going to be spending a lot of time with the PDB over the next couple of lectures that I give you. And finally, we learned a little bit about sequence motifs. So on that note, are sequence motifs more likely to be conserved between monkeys and humans or monkeys and birds? Is this as straightforward a question as you think it is? In some cases, yes, because we know that monkeys and humans are more closely related. And so it's more likely that monkeys and humans will share more common sequence motifs. On the other hand, there are some very basic functions for life at the vertebrate animal level, of which birds and monkeys and humans are all part of vertebrata. And so there are probably a lot of sequence motifs that are extremely important for regulating different functions that are much more basic than simply the little differences between monkeys and humans. So think of regulating breathing, for example, or regulating body temperature. Vertebrates, warm-blooded creatures, birds and animals are both warm-blooded. They will have certain proteins that participate in the regulation, in the thermal regulation, or the maintaining the metastasis of the body, so maintaining the general consensus happy state for the body. A lot of these things are done through regulatory mechanisms, and the proteins in those regulatory mechanisms are very likely to maintain surprisingly similar sequence motifs. So the correct way, if there is one, to answer this question is that it really depends on the kind of motif you're looking for. If it's something specific between monkeys and humans, for instance, the proteins that control some of the fast twitch fibers in the face to show emotion, then monkeys and humans will certainly have more sequence motifs than monkeys and birds. However, if it's something like I mentioned, like breathing or thermoregulation, or if it's just the regulation of cell division, something that's very basic, then monkeys and birds would still have a lot of related sequences and sequence motifs. Okay? So this concludes my presentation. I wanted this lecture to be a little bit more theory intensive. And again, I apologize if some of this stuff went over your head. But I figured that we needed a balance from last week's demonstration bonanza that I gave you. I highly recommend that you email me as soon as possible with any of questions you may have for any of the lectures I've done up to this point, and let's make sure that we are on the same page before we dive into protein structures. Okay, well, have a nice weekend, and do be in touch and let me know if I can help you out with anything. Right, take care. Thanks very much.